Okay, it is 1:30. Exam one is posted, or at least it should be. It became visible to the students yesterday at noon. It's due Thursday by noon. It covers chapters 1 through 5. You have to take it with Preptorio. I am leaving for Phoenix, Arizona on Saturday, and I'm coming back on Monday. Monday and Tuesday of next week are holidays. And so I won't be back on campus until Wednesday. And so if there are all kinds of problems with exams or anything like that, I'm not going to be able to do anything about it until a week from tomorrow. Um, and I think the exam is ready to go. I have given some people time and a half. Most people have 50 minutes to take the exam. If you have time and a half, it extends you to 75 minutes. And I advise not waiting until the very last minute to take the exam. Try to take it, say, for example, tomorrow. And if you run into problems tomorrow, that gives us, well, 24 hours to fix anything. Now, although this meeting is scheduled to go until 2:45, I at most can stay an hour because I have another meeting to go to at 3. And that's going to take until 4:30, 5 o'clock. And I'm going straight home after that. I'm not coming back to the office. Okay, this is chapter 6, which is basically extranuclear inheritance. And it's got some other things, some fairly complicated things called imprinting. And then there's a very bizarre kind of um, observed inheritance called maternal effect. And again, in simple Mendelian inheritance, you have basically in simple Mendelian inheritance patterns, there's one physical trait that is encoded by one gene. And at this gene, there are two alleles. And one of those alleles is dominant, one is recessive. And we say that in Mendelian inheritance, the inheritance patterns follow the laws of segregation and independent assortment. And Mendel himself did not take into account the fact that mutations might occur. Mendel himself did not look at genes for a long enough period of time to observe any mutations. And when it says expression of the genes in the offspring directly influences their traits, it means that there is no environmental effect. Your genotype determines your phenotype. Now, it says some genes violate these rules. Most genes violate those rules to some extent. But there is some very definite non-Mendelian inheritance patterns. And a couple of them are easy. Some of your organelles have their own genomes. And typically we just say it's the mitochondria and the chloroplasts. They don't have many genes, but they do have genetic material. And then there's something called epigenetic inheritance. Genes can get altered. For example, you might methylate chromosomes. You add methyl groups to them. And methylation typically makes the chromosomes more compact. And methylation typically shuts down the expression of genes in the area of the chromosomes that has been methylated. And then there's maternal effect, which is the genotypes of the offspring don't determine the traits, but it's the genotype of the mother determines the physical traits in the offspring. And there's an example of maternal effect, and we get to that near the end of the chapter. Now, this is extranuclear inheritance. It's easy to explain. And we're going to start off with chloroplast. And so plants and algae are going to have chloroplasts. And any gene that you refer to as a nuclear gene is a gene on a chromosome inside your nucleus. Extranuclear inheritance is something that you inherit from some other organelle. And they say it's cytoplasmic inheritance. I've never seen cytoplasmic inheritance other than in this textbook. And the two most important examples are genes that are found in mitochondria and genes that are found in chloroplasts. And like I said, there's not a whole lot of genes in the mitochondria or the chloroplasts. In human mitochondria, the number that is repeated is in human mitochondria, there are 37 genes. Chloroplast genomes are about 3 times that size, 100 and something or so. And so here is the chloroplast genome. It is circular. It resembles the chromosome that you would find in a bacteria. Now, there might be multiple copies of this chromosome in the organelle, but they're all the same. Mitochondria and chloroplasts do not have nuclei, but they have this nucleoid region where you find the organellular genome. There also might be multiple nucleoids, each with multiple chromosomes in the organelle in question. And there's an image of mitochondria, and the nucleoids have been color-coded in yellow. And the genetic composition of chloroplasts is, well, it's kind of species dependent. But you have these green algae. There's a green alga called a chlamydomonas in the chloroplast of that algae. There's about 15 or so nucleoids per organelle, and there is about 80 or so chromosomes per organelle. And then you have another type of alga, a euglena, 20 to 30-ish nucleoids per organelle, 100 to 300 chromosomes per organelle. And then the typical flowering plant, a dozen to 2 dozen or so nucleoids, and about 60 total chromosomes per chloroplast. And so you abbreviate chloroplast genome with lowercase cp. Main function of a chloroplast is photosynthesis. And so most of the genes in the chloroplast genome are genes that encode proteins that are involved in photosynthesis. And a fairly well understood chloroplast genome is that found in tobacco. And it's about 150,000 base pairs, 110, 120 or so genes. These genes include sequences that encode RNA and tRNA. And it's got some genes that encode the mRNAs for the proteins necessary to do the reactions in different types of photosynthetic processes. Now, a lot of the proteins used by the chloroplast are encoded by genes in the nucleus. It is believed that once upon a time, these proteins were encoded by genes in the genome of the free living bacteria that got engulfed and eventually became a chloroplast. But over the last couple of billion years or so, since this happened, a lot of those genes got moved into the nucleus. Now, In chloroplast inheritance, well, you don't get your chloroplasts, or for that matter, your mitochondria, equally from both parents. You typically get your organelles from one parent or the other. And it is usually you get all of your cytoplasm, which include all of your organelles except for the nucleus, from your mother. But that is not going to be 100% true. And so these organelles are not evenly divided up during meiosis, and therefore they don't go into the gametes like the chromosomes in the nucleus do. Now, there is a type of plant, Mirabilis jalapa, and its leaves can be green or white, or they can be mottled. And the pigmentation of that leaf is determined by the chloroplast. And so basically, in this plant, green leaves are wild type. And they have normal chloroplasts that make a green pigment. The white leaves are mutant. And in those chloroplasts, they don't make that green pigment. And in the plants that have mottled green and white leaves, you have inherited in those particular cells a mix of wild type and mutant chloroplasts. And so in the case of these mottled leaves, both parents contribute chloroplasts to the offspring. And we call that heteroplasmy. And so, 1909, leaf pigmentation was shown that it could not be predicted by Mendel's two laws. And so they said that is a non-Mendelian pattern of inheritance. And usually, the pigmentation of a leaf depends upon the genotype of the mother. So, in most cases, pigmentation is influenced by the mother's chloroplasts. And so, basically, you can say simply, pigmentation depends upon the mother's phenotype because the egg provides the cytoplasm. Paternal pigmentation has no effect because the sperm provides no cytoplasm. In most species, the egg is a gigantic gamete in comparison to the sperm cell. And so the egg contributes the nuclear DNA plus all of the cytoplasm with all of the organelles. The sperm usually contributes only 1/2 of the nuclear genome. And so basically, it's pretty simple. If you have cross number 1 with a white male plant and a green female plant, well, all offspring, white female, green male, all of the offspring will be white. You do the reverse. Green female, white male, all of the offspring should be green. And then you do this experiment cross. A variegated female plant, green-leaved male. Well, you have the opportunity to get green or white or variegated offspring. And if you set up the reciprocal of the above, you should get all green offspring. That's the typical prediction you would make. And so in this particular plant, the inheritance of the variegated phenotype is a result of the way the chloroplasts are segregated. Chloroplasts during cell division get randomly segregated into the daughter cells. And so if a plant develops from a zygote that happens to have mutant and wild type chloroplasts, just by chance, some cells will get all or mostly wild type chloroplasts. And you will see the regions of those parts of the leaf as green. Some cells will get all or mostly mutant chloroplasts, and you will see those as white. And some cells will get about a 50-50 mix of wild type and mutated chloroplasts, and you will see that as green. And so typically we say, in these non-pigmented areas, they have all mutant or mostly mutant chloroplasts. In these green areas, they might have some chloroplasts carrying the mutation, but they're going to have at least some that are wild type and encode enough green pigment to make you observe that thing as green. And so, like I said, in chloroplast inheritance, usually they go through the maternal line. And it's easiest to think of what we call a heterogamous species. The male gamete and the female gamete are very distinct from each other. And egg cells tend to be big. They're packed full of cytoplasm. They're filled with organelles. A sperm cell is basically a nucleus with a flagellum. Now, there are some cases where this is not true. Gymnosperms, for whatever reason, the chloroplasts are inherited through the paternal line. And for some angiosperms, for some flowering plants, the chloroplasts are inherited basically equally from mother and father. And so for extranuclear inheritance, you have to know a couple of things. Which parent contributes the organelle in question? And if you have this heteroplasmy, you kind of have to know, if the parent has heteroplastic phenotype, you have to know where that gene might be contributed from. Again, boils down to which parent might contribute it. And again, in algae, the chlamydomonas, you have the chloroplast inherited from a parent with a specific mating type. And in this type of alga, there's MT plus and MT minus mating types. It's not two different sexes. It's two different mating types. And the individual with MT plus contributes the cytoplasm. In flowering plants, usually it's maternal inheritance, although there are exceptions. And in gymnosperms, chloroplast inheritance is almost always through the male parent's line. And then you have genes in the mitochondria. And again, features of the mitochondrial genome, how to predict crosses, and talking about some mutations in some of the genes in the mitochondria. And it's kind of similar. Mitochondrial genome is circular. It's contained within a nucleoid region. There might be multiple copies of that chromosome within a given nucleoid, and there might be multiple nucleoids in any given mitochondria. And so in tetrahymena, which I want to say is a type of fungus, but don't quote me on that one. You have the mitochondria that has a single, typically has a single nucleoid, and there are 6, maybe 8 copies of that chromosome in that nucleoid. In a mouse, you might have 3 nucleoids. And in a single mitochondria, you might have up to about 6 total chromosomes. And again, the nucleoids have been colored in this image. Okay, main purpose of a chloroplast is to carry out photosynthesis. Main function of mitochondria is to undergo oxidative phosphorylation to make ATP. And the mitochondrial genome is abbreviated lowercase mtDNA. Now, in humans, the mitochondrial genome is very small, 17,000 bases. It has a few genes. Typically, we say 37. Some of these genes encode RNAs and tRNAs that function within the mitochondria. 13 of them or so encode mRNAs that get translated into proteins that work in the mitochondria. And again, just like the chloroplasts, most proteins that work inside the mitochondria are encoded by genes inside that organism's nucleus. And again, just like in chloroplast, we think once upon a time that bacteria that was engulfed, that eventually evolved into mitochondria, a lot of those genes from that bacterium are now present in the nucleus of the cell in that organism. And this is the human mitochondria. It's been mapped. And again, it's got some rRNA genes in it. And it's got some tRNA genes in it that carry amino acids to the growing polypeptide chain. And then the color code. Well, here's a piece of random junk DNA. And the other ones encode things that are usable or useful in making various components of aerobic respiration. And so you have a couple that encode ATP synthase. You have a few of them that encode NADH dehydrogenase and so on and so forth. Now, like in chloroplasts, mitochondria are almost always, but not necessarily, inherited through the mother. And so in mammals, it's almost always maternal inheritance. In this particular yeast, it's biparental. In other fungi, it is usually maternal, but there are some weird exceptions. And in algae, mitochondria come from the parent with the MT minus mating type. Chloroplasts come from the parent with the MT plus mating type. And then in flowering plants, it is usually maternal inheritance, although biparental inheritance has been observed. And in gymnosperms, it's usually paternal inheritance. Now, every now and then, I want to say that every now and then there is some quote unquote leakage. And so in mammals, usually you get your mitochondria from your mother, but every now and then a sperm cell might contribute a mitochondria or two, but they're vastly outnumbered from the mitochondria from the female. Now, again, there is every now and then you find quote unquote leakage, which is mitochondria provided through the sperm cell. And so they say, for example, in the mouse, 1 to 4 paternal mitochondria are inherited for every 100,000 or so maternal mitochondria per generation. So it's a very small number. Now, there are documented human mitochondrial diseases, and there's at least two types. One is zygote might inherit a mutated mitochondria from the mother, and these are easy to trace. Almost always through the matriarchal line. Also, you can, as you age, you can have mutations occur. Mitochondrial DNA is more prone to mutation than nuclear DNA. The mitochondria do reactions that generate byproducts that just have an ability to damage DNA. And mitochondria do not have the world's best DNA repair capabilities. And so DNA in the mitochondria is just more prone to error. And there's been at least a couple of hundred mitochondrial diseases known. And they're usually found in cells that need a lot of ATP. And for example, they're typically found in muscle and nerve tissue. And there's a list, there's a sum of them. And it's almost always on here some kind of neuropathy, nerve damage, or myopathy, muscle damage. And there's a few different mutations that can contribute to, various diseases. This isn't a genetics of disease course. So this is trivia. Okay. And this, again, should be a review, although I must admit it is only mentioned in Biology 1. It's the endosymbiosis theory. And the explanation of the endosymbiosis theory is that mitochondria and chloroplasts, a couple of billion years ago, were free living bacterial cells. And it says that chloroplasts originated from a cyanobacterium, a photosynthetic bacteria. And we think that mitochondria came from a gram-negative, non-sulfur purple bacteria. And I am not a microbiologist, so I don't really know the importance of it being gram-negative, non-sulfur purple bacteria. But those bacterial cells that got engulfed eventually evolved into the modern mitochondria or chloroplasts. And there's the idea. And so here is a lineage of cells that's on its way to developing into what we would nowadays call a plant. And here is a lineage of cells that was nowadays on to what would evolve into something we would describe as a non-plant animal or a fungus. Plants have both of these things. Animals and fungi only have mitochondria. And so here's the idea. And there's a lot of reasons this idea gained traction. Individual mitochondria and chloroplasts are about the same size as bacteria. The genomes are circular, like in a bacteria. If you sequence the organellular genome, the DNA looks prokaryotic. You can tell. You can tell the difference between prokaryotic DNA and eukaryotic DNA. It has a fairly obvious advantage to the host cell. It allows that host cell to do photosynthesis and make more ATP. Well, it does both, or just that second one. It's more difficult to explain how the bacteria that were engulfed benefited much, other than basically they now had a host cell in which to live. And during the last couple of billion years, most of those ancient bacterial genes has basically mutated into non-functionality, or they have migrated to the nucleus. And that lateral gene transfer has been almost 100% in one direction, from the organelle to the nucleus, and not the reverse. Okay, that's a good place to stop. That's about, more or less, believe it or not, halfway through the chapter. I plan on finishing chapter 6 Thursday. I'll go extra time if I need to. And I'm leaving town on Saturday. I'm coming back Monday. It's probably the last time I'll see my father before he dies. I get back on Monday. School's closed Tuesday. It's Mardi Gras. We reopen on Wednesday. And basically, that's when I'll be back to handle anything, is a week from tomorrow. So, like I said, I'm going to stop there because I have another meeting in a few minutes. And I need some time to prepare for the second meeting.